Thanks very much, everyone, for coming along. So Opteran is a natural intelligence company. It's a company that is based on decades of trying to understand how real brains work and reverse engineering that in order to come up with new approaches for machine autonomy, for robots and other autonomous systems. And every year we recognize the pioneers in the field of natural intelligence, the people who have helped develop this insight and blaze the trail for us and others to follow in understanding how the brain truly works. This year, we're very happy and honoured to be presenting to Professor Andrew Barron. So Andrew is at Macquarie University in Sydney, but he started out in the UK, he's actually British, and studied at Cambridge for his degrees, and then went to postdocs in America and Australia before finally settling down there. He's a pioneer of many things, and we've had a long-standing relationship. He's also visited Sheffield with two different visiting professorships from the Royal Society and the Leverhulme. So long-term collaborator. And he's among his many hats, he is what's called a comparative cognition researcher, which means he'll explain what that means, but it briefly means he tries to understand how different brains solve the autonomy problem and the commonalities and the differences between those. So Andrew, as one of the pioneers of natural intelligence this year, we are very happy to present to you the Opteran Mind Award for 2024.
Brilliant. Thanks, James. Put this down there safely. Great.
Thank you.
James, thanks very much. I'll get right into it. It's always nice to be back in Sheffield. I had 2 summers here with my husband while we were working with bees at the university here, and it really struck us it's such a beautiful city. It's such a really vibrant university, so it's really, really nice to be back here with you today. And today I'm going to be talking about how honeybees think and how I think the honeybee mind works. And that was making me realize that I'm now really comfortable talking about a bee as something that thinks and a bee that something possesses some kind of mind. But it hasn't always been that way. When I was an undergraduate, I was really influenced by the work of Vincent de Chia. And one of Vincent de Chia's quotes that really struck me is, "The fly is colossally stupid." Now, I really want to say that Vincent de Chia was an absolute pioneer of insect neuroscience and insect comparative physiology. And he didn't make this as a flippant comment. This was based on decades of work trying to train flies to do something. As someone who spent their PhD trying to train flies to do something, I am sympathetic with this comment. But it's also interesting how even back in the 60s, the field was changing and perceptions of insects were changing. So just 2 years later, back in 1964, de Chia wrote this beautiful article in Science, and one of his quotes that really struck with me is a pondering about what insects might be. Are we thinking of insects as some little robotic automaton, or is it something more? Is there someone inside? And that's actually my PhD thesis. It's a lovely font, thank you. And this quote really stuck with me, and I've realized this kind of question of what is an insect has shaped most of my research that's gone on since my PhD. And of course, the field of comparative cognition, and particularly insect cognition, has moved on in leaps and bounds. So we now are very comfortable talking about the scientific research in bee cognition. There have been pioneers in that research here with us. And I have this as a list of the claims that have been made about what bees are able to do. And it's a spectacular list. Each of these raises a question about if bees are doing this, how are they doing it, what are they really doing, what kind of processing is going on in their brain that enables them to do these kind of things. But we would not at this point deny that a bee is cognitive. A bee is a cognitive organism. What I've come to realize is the nature of that cognition has been enormously shaped by the demands on the bee's lifestyle. So the bee is a central place forager. It does a job that would be impossible for me to do. It makes a profit, an energetic profit, gathering pollen and nectar from flowers. So it's navigating across kilometers of distance. It's finding cryptic, ephemeral resources that change their distribution moment by moment, day by day, hidden in minute amounts across an enormous array of flowers. It's making moment-by-moment economic decisions about whether to persist in foraging or whether to try moving on to somewhere else. And it's making a profit. I can't even find Greg's in the morning for a coffee with Google Maps. And yet the bee is solving all of this with a brain like this, a brain that is absolutely minute. So to scale you in, this bar at the bottom here is 1 millimeter. So we have the insect brain, this millimetrically scaled structure that is packed into and fills the bee head, containing about 1 million neurons, which compared to us is not much, but 1 million is not trivial. So it's a small brain, but it's not a simple brain by any means. And what really stands out about insect brains and what makes them wonderful to study is they are so beautifully modular. And the structure-function mapping in the insect brain is exquisite. So the insect brain made up of multiple modules, each with very clearly specialized roles, each with very clearly different computational architectures in different regions. We have lobes that are specialized for the processing of sensory input. We have sensory integration centers that combine that input for different functions. And one of the most important sensory integration areas that I've worked on a lot, and that also dominates the shape and structure of the honeybee brain, are these regions here called the mushroom bodies. So this is a contemporary micro-CT scan, three-dimensional scan of the mushroom bodies in the honeybee brain. This is a light micrograph drawing by Félix Dujardin in 1850. So it's not just that we've got a comprehensively scaled brain, we've also got a really long history of detailed anatomical, behavioral, and electrophysiological investigation of this structure. So thanks to that, I think it's fair to say, certainly per neuron, we know a lot more about how an insect brain works than we know about how the human brain works. This mushroom bodies, for example, these take up about 250,000 neurons in the bee's head, so 1/4 of the bee's neurons are packed into this structure. We now recognize that it's a 3-layer structure with a layer that forms the cup of the mushrooms here that receives multimodal sensory input. A second layer makes up the body of the mushrooms, the so-called peduncles, a large population of 250,000 neurons. And then there's a third layer at the base where they output into the premotor regions of the bee brain. And for the computer scientists in the room, that kind of mostly feed-forward 3-layer structure we would recognize as a motif that is very common in deep learning, machine learning, and perceptrons. And it's recognized in computational science, computational neuroscience, as this kind of structure is extremely good at solving classification-type problems. And I want to highlight some of the work that was done here in Sheffield by Alex Cope, who is here, Eleni Vasilaki, and James Marshall, basically using this kind of modeled understanding of the structure of the mushroom body to really deeply probe just how complex a classification can this structure solve. And this is a really beautiful example, I think, of where a biorobotic and computational modeling approach let us interrogate how the mushroom body might be working, with the answer being a whole host of complex classification problems can be solved by a structure like this. And classification is really core to the kind of challenges that bees face in their everyday life. This is the kind of problem a foraging bee faces, a field of mixed flowers. And it's not just a matter of a bee identifying which species of flower is the one most likely to offer pollen and nectar. It's also got to identify exactly which age, which stage of flower marks it as being the ones most likely to offer nectar, in what condition, at what time of day, etc., etc., etc. So a complex, multi-part, multimodal classification problem. And we know from their behavior that bees solve this extraordinarily well. And I wanted to kind of test the bees' capacity as classifiers of complex information. So this wasn't an experiment, it was a demonstration. I wanted to challenge myself whether I could train a bee to classify and to recognize different styles of impressionist art, whether the bees could do it and how long it would take them to be able to do that. So here we have 3 impressionist painters, Émilie Carr, Claude Monet, and Georges Seurat. So all impressionists, all landscape painters. So all the paintings have the same kinds of tones and the same kind of subject matter, but clearly 3 very distinctive styles. The question is, can a bee learn to classify a painting by its artistic style? Now, the way you do an experiment like that with a bee is you start off getting the bee to pay attention to the task. So first of all, actually, the first thing you do is you mark the bees so you can tell who's who. So you stick a numbered tag on the back of the bee so I can tell who I'm working with. And the first thing we did was to train the bees to attend to nice, simple yellow squares. And the way you do that is you put a drop of sugar water onto a little yellow square on a little card, so the bees rapidly learn that if they go to a yellow square, they will get a nice sugar reward. Then having done that, we set up in a nice flight arena what we call a conditional discrimination task. So we wanted to train them to recognize painting by artistic style. So at the entrance to the arena, we set up painting by one of the artists. Inside the arena, we set up an array of different paintings. We had to train them to pick a different painting by the same artist. So to do that, we put sugar on those paintings. On the other paintings, we put a drop of quinine. Now, quinine is not harmful to bees, but it tastes bitter. So the idea being, if they landed at the wrong painting, they would get a slightly bitter taste. If they landed at the right painting, they would get a sugar reward. And then over successive trials of this, we're hoping that we should build in the bee them learning to match paintings with other paintings in the same classification, in the same category. So this is a training trial. So in this training trial, we've got a painting by Claude Monet at the entrance, which means in this training trial, on the Monet paintings, I've put sugar water. On everything else, I've put quinine. So this bee's seen Monet in the entrance, but she's still learning. I want you to watch her behavior as she comes into the arena. So for the bioroboticists in the room, these beautiful scanning flight paths, moment by moment evaluations and decisions of what to do. And here she is taking a good look at all the paintings. Now, see, she's touched an Emily Carr painting. It's bitter. She rejects it and flies away. That's my bad camera work. And then she lands on a Claude Monet. It's sweet. She lands, is fed. She'll feed to repletion, and then she would leave the arena. That completes one training trial. So we gave her just 18 training trials in just a couple of hours in one afternoon. And then in the test, again, she's going to see a painting at the entrance. But in this case, we've put sugar on everything. So in the test, the question is, having seen this Emily Carr painting at the entrance, will she go directly to an Emily Carr painting? And this is how this test looked. So she comes in, she investigates the painting at the entrance and inspects it, enters the arena. Now again, you get this beautiful scanning behavior, but notice she's not landing yet. She scans all the paintings that are available to her. That's an Emily Carr. She lands and she feeds. So 18 training trials, and this bee has learned to match painting by artist. Now, I wanted to make it more difficult, because of course she could have learned to match this specific painting to that specific painting. So we took 3 paintings she had never seen before in training, 3 new paintings by the same 3 artists, and with no additional training, we asked, could the bee be successful in matching one of these paintings according to its artist? So here is that test. So here's an Emily Carr she's never seen before. Now she lands on it, which is interesting. So there's something that's confusing her about this, I think. He says, be anthropomorphic. She comes into the arena, again, inspection without touching, inspects all the painting, and lands on an Emily Carr. Again, she inspects the Emily Carr, comes into the arena. I'll play this one more time so you can see it, because it cut too quickly. Inspection without landing, and lands on the Emily Carr. So even a painting she hasn't seen before, after just 18 trials in this task, she's now able to classify painting by artist. Now, what struck me by that, and this has been striking me for a while in my research, is the speed at which bees learn these kinds of classification tasks. And whenever we compared our literature, when I was talking at conferences about this, we always see bees learning in like 30 trials to 60 trials, these kind of quite complex classification type problems. If we compare that with other animals, it does seem like the bees are learning unusually fast. So this is work by Quinn Solvey. It's work in progress. It's not published, but I'm sharing the data with you here. We kind of did a meta-analysis going back through the extant literature of all these kind of rule learning tasks, where it's like, if A, select C, if B, select D. Tasks with that kind of structure. And we asked, how well did the animal do out of 100, how many training trials did it get for them to reach that peak level of performance? All the bee studies that have been done form this kind of cluster down here. So all the bees do about like 75% above chance, pretty good. And they take about 30 to 60 trials to do it. For all the other animals that were studied, rodents, primates, a dolphin, birds, you get the same kind of level of performance. It's at least comparable. But this is a log scale. They're taking hundreds to thousands of trials to learn. So again, it reinforces this impression that bees are unusually fast in learning this kind of classification task. But in none of these studies did you ever have an explicit bee other thing comparison. So we tried to do that. We decided to try and compare learning in one of these conditional discrimination tasks between honeybees, bumblebees, and hummingbirds. And we picked those three because they're all organisms that have all evolved to extract food from flowers. So they've faced the same kind of ecological challenges in their cognitive evolution. And also they're all highly visual organisms, they have good color discrimination, and they're all motivated to select little droplets of sugar from their environment. These are the tasks that we set them. Again, it was a conditional discrimination. So in this case, in the bumblebee example, if they saw white here as they entered the arena, for example, they had to pick blue to be rewarded. If they saw black here, they had to pick yellow to be rewarded. This is the same task. It was a slightly different shape and size of the arena for a honeybee, but conceptually the same task. For the hummingbirds, it looks a little different. They don't like white and black as much. They respond much better to yellow versus red. And they don't really like the horizontal arrangements. They prefer vertical arrangements, and they need perches. But conceptually the same task. So we adapted it to suit the sensory system of the organism and the physical limitations of the organism, but conceptually the same task. And these are the data that we got. And again, we saw our two bees learning the task within about 30 trials and getting to maximal performance. The hummingbirds took an awful lot longer, 80 plus trials, to reach a similar level of performance. Now it's work in progress. Our working hypothesis is that counterintuitively, it's at least possibly feasible that a small biological brain, as long as it's big enough to do the job, might be faster at learning this type of task than a larger brain. Perhaps because there are fewer pathways, fewer modules that need to be adjusted and adapted in learning this kind of path, this kind of task. Now, there's almost certainly a trade-off to that. It's also possible that a bee brain with its million neurons might have less capacity for numbers of different things it can learn than the 200 million neurons in the hummingbird brain. And there might be this nice trade-off, perhaps, between your learning speed and your learning capacity. But if you're an organism like a bee, which doesn't live very long and needs to learn quickly in order to be an effective forager, it might be more important that you can learn fast than having a very, very large capacity of the different things you can learn. But the one thing that we can say is that this bee is not colossally stupid, not by a long shot. Now, for the second part of the talk, I want to come back to the second quote by Vincent de Chier and the work that's really consuming me now, which is this question that Vincent de Chier posed, which is, how do we think about an insect? What's the best way to describe what an insect is? Is it an automaton? Is it robotic? Or is it something more? Is it something conscious? Is it something experiential? And that's a question that I posed with Colin Klein back in 2016. I mean, with Colin, we wrote this paper, What Insects Can Tell Us About the Origins of Consciousness. And at the time, that was a deliberately provocative paper that we wrote, not with an answer, with a question, to pose a conversation about this, about how should we think about insects. And what we were proposing is that we were arguing that it's not inconceivable that an insect could have perhaps the most basic type of conscious experience, conscious capacity, that an insect might be something we could describe as experiential. The idea of this work was that it started a conversation, it started a debate. And that certainly did trigger a debate, and it was part of many things that were triggering a debate at this time. And very recently, we've seen, in fact, this new declaration on animal consciousness that came out of New York University, led by Jonathan Birch and colleagues. And in that, it's a very, I think, very sensibly and very cautiously phrased update of a declaration on animal consciousness that at least raises the possibility that based on the experimental evidence, there is now at least the realistic possibility of some form of conscious experience in some invertebrates, including at least crustaceans and insects. So cautiously phrased, but raising that we at least have a realistic possibility of there being some sort of conscious experience in insects. Now, this declaration took a weight of evidence approach. What it was really asking is, based on all the evidence that we've now accumulated from all of this research into physiology and neuroscience and cognition, how has that moved the dial on whether it's likely or unlikely that an insect might have some sort of conscious experience? And the kind of evidence that they were discussing were things that we could consider to be markers of some sort of conscious processing. So things like evidence for anxiety-like states, complex motivational trade-offs that subjectively change the organism's behavior, sleep and active sleep, and social facilitation of different types of sleep. There's now evidence for all of these things in invertebrates, particularly in insects. Now, this marker approach is really good and very, very appropriate if the question that you need to address is, how likely is it that we could have some sort of conscious experience in an insect, likely or unlikely? But with a marker approach like this, it really does kind of collapse the question into a whole load of questions about the markers. Are the markers good or not? Each of these markers could be challenged, and someone could pose, well, for any one of these, we could imagine a system that could be entirely robotic, but display this type of behavior. So the weight of evidence becomes persuasive, but each of the markers becomes a series of sub-questions about what's plausible, what's a good marker, what's a bad marker, what do we do if an organism shows some markers but not all of them. For me as a neuroscientist, what interests me is that if we're saying that it's plausible an insect has consciousness, the most interesting question for me is that then how and why is an insect conscious? How would consciousness work in something like an insect brain? And that's the question that I'm devoting my time to. That's the question that I'm enjoying getting obsessed about. And I want to really caveat at this point that it's important to realize that when we're talking about the kind of level of consciousness that might be possible for an insect, I'm not saying the insect is experiencing the world and thinking the way that I experience the world. Consciousness is an extremely complicated phenomenon. It is multi-component, multi-ambiguous, and highly complex with multiple different types of conscious experience. What I'm imagining is that if anything, an insect has what we're calling phenomenal consciousness, which many would consider to be the most basal form of consciousness possible, distinct from self-awareness, distinct from meta-self-awareness. Phenomenal consciousness simply describes a system that we might describe as being experiential. So there's if something has phenomenal consciousness, there's something it's like to be that thing. Just as we might imagine there's something like it's like to be me or my dog, there's not something it's like to be a rock. So the capacity for phenomenal consciousness, there's something it's like to be if you have a capacity for phenomenal consciousness. You're experiential. Now, in my thinking about this, I've been really influenced by the work of Bjorn Merker, who studied humans and other mammals. And Bjorn Merker localized the capacity for phenomenal consciousness to the human subcortex and midbrain. Now, these would be considered by a lot of people the more primitive regions in our human brain, so subcortex and midbrain. And Bjorn Merker localized phenomenal consciousness to these regions for all sorts of evidence, but one of the lines of evidence he tapped into came from the work of Wilder Penfield. And if you want a good read, this book, Mystery of the Mind, is an absolutely brilliant read into experimental neuroscience. Wilder Penfield was a pioneer in methods of surgery, and he developed a way that he could operate on the brains of conscious, awake, responding participants. Now, that surgical technique was developed because he was trying to treat extremely serious and recalcitrant forms of epilepsy. So the idea being that you would try and actually cut out of the brain the epileptogenic center in the brain that was triggering epileptic episodes. And the only way that you could do that is to operate on conscious patients as you were, I wouldn't want to do it, carving away at the brain, cutting out the bit that you think might trigger epilepsy while leaving all the good bits intact, and hope you didn't go too far. But what Wilder showed in his work was this is the extent of cortical regions you could cut away from an intact, conscious responding person without actually in any way interrupting their conscious stream. So based on that and other evidences that Merker draws on, Merker argues that for phenomenal consciousness at least, it must be localized deeper in the brain. Now conceptually as well, Merker had argued that the reason that we would expect phenomenal consciousness to be localized to the deeper, more primitive regions in our human brain is because the core function of the brain, which localizes to the midbrain and to the subcortical structures, is to generate an action command. The core function of the brain is this problem of solving autonomous decision-making. What the brain needs to output is some sort of coordinating, organizing command to the body that organizes it to do one thing at one time. And the job of the brain is to identify what that one thing should be. And to do that, this problem of solving autonomous decision-making, Merker identified there's 3 real different domains that each need to be resolved alongside each other to solve this problem of autonomous decision-making. You've got to identify what your objective is. You've got to identify your target. What do you need to achieve or obtain to meet that objective? And you've got to identify the action that you've got to perform to get the target to meet the objective. But you can't resolve each of these domains of autonomous decision-making in isolation because they all interact. So the array of available targets is going to influence which objective is the most important right now. And the objective most important right now is going to influence which of the possible available targets is going to be the one that you hit. The target is going to influence which action you would take to get it, whether you've got to take a bus or whether you've got to just walk across town. But the ranges of possible actions also, and the potential costs of different possible actions, that's going to influence again which targets you're going to hit. And the array of possible actions is going to interact with which objective is feasible and which objective is the priority right now. So these different domains of autonomous decision-making have to be able to interact. And Merker recognized that the mechanisms for each of these domains of decision-making, they all localize to the more primitive, fundamental, basal parts of our brain, the midcortex and the midbrain and the subcortex. And we've got a whole list of absolutely critical different systems in the brain that contribute to each of these domains of decision-making. So what we've got is a system of systems, an interface to solve the problem of autonomous decision-making in a biological brain. And Merker was very big and very heavy on the neuroanatomy, which I think makes Bjorn Merker's work sometimes quite hard to read. I'm not going to dwell on it too much, but just it is worth, if you know human neuroscience, it's really interesting to read Merker's perspective on how the brain works. He highlighted the superior colliculus, which is located here in our brain, as being, if you like, a reference framework that takes all spatial information from any sense. Once it's been processed and corrected for self-motion, I know that correction for self-motion is still a challenge in robotics and one that Opteran's been making great progress to solve. But all that information is put into the same kind of spatial reference, spatial framework that then becomes a unified framework for resolving the problem of targets, which targets are available and how are they localized and arrayed around me. He also highlighted the importance of the hypothalamic nuclei, the nuclei of the thalamus, the I'm having a brain fart. And the nucleus of something else. Thank you. as being important for resolving the objective functions, for resolving different potential competing motivations to identify what your objective would be. Thank you. Glasses. Welcome to aging, people. Hypothalamic nuclei, nuclei of the thalamus, nuclei of the basal ganglia. An integrated system, and I know there's people here who've worked on basal ganglia very, very much, but integrated system of systems that can resolve problems of competing motivations and process information on the internal state of the body to identify which of the objective functions is the one that should be primary. And again, these regions, if we look at a section of the brain, they kind of localize here, deep in the midbrain, and if we take Michelangelo's drawing of the brain, they localize here into kind of God's crotch, which is a little bit awkward. And then if we think about the system of systems in totality, we have an interface that lets these systems that organize space and targets available interface with systems that organize objective functions and motivations, so we can get this resolution of autonomous decision making. And Colin Klein and I are calling this interface a phenomenal interface. And we've called it a phenomenal interface because a consequence of the way in which this system works is that it also an outcome of this of a lot of features that are actually really quite important for the claim that the entity operating an interface like this is in fact experiential. So a consequence of processing information in this way is that the outcome of processing information in this way is that you have a coherent, unitary, and organized unitary sensory percept. Everything is in one common spatial frame. All spatial information feeds into the same sensory percept. That sensory percept is organized from a singular perspective, a singular point of view. And that point of view has the properties of kind of an ego center, because your possibilities for action, your possibilities to perception, localize to a singular point that is localized within the body of the organism that is processing the information in this way. So it conveys on the organism a first-person perspective that distinguishes the organism from its world and gives it a perspectival view on the world. The information processing also becomes subjective because it's completely tied up to the state, condition, and point of view of the agent that is perceiving all of that information. So the information becomes subjective. And of course, the system also supports the autonomous decision-making of the organism, so it conveys on the organism a capacity for agency. So these are all features that I think are really important in saying, well, if it's demonstrating all these kind of things, the best way to talk about something like this is something that is experiential. So I think we can start to say that we've got a system that is experiential. Now, Bjorn Merker was talking about the mammalian brain and the human brain. I think this is the abstracted element that's key to what Bjorn Merker was saying. This kind of phenomenal interface. And what I would argue is that when I look at how information is processed through the insect brain, I see the totality of the insect brain essentially operating as a phenomenal interface. What we have in the insect brain are an integrated system of systems, an interface of systems that works together to resolve the autonomous decision-making in the same kind of way as this phenomenal interface. I'm not going to go into all the detail here, but when we break it down in terms of the modules of the insect brain and how I think they interact, I think it's fair enough to describe it as an insect version of a phenomenal interface. And for that reason, I think it's fair to say that we can make a claim that an insect is at least experiential of the world around it. Now, this is, of course, still a hypothesis. It's not completely resolved neuroscience by any means. But I'm really pleased to talk here today because I think the challenge to address that hypothesis and to test this hypothesis, we're going to need to lean really hard on computational modeling and biorobotics, because we're going to need the tools of computational modeling and biorobotics to actually help us pose the questions and test the hypotheses that we're putting forward here. You know, we've got skin in the game here. We've made a lot of progress in resolving the capacity of the mushroom body by taking it into a computational modeling perspective. You can do things in computational modeling that are wonderful interventions where you can lesion, and Alex Cope did this beautifully, you can lesion selectively different elements of the system to ask the question, how is it really working? And you can do it in biorobotic applications with far more precision than you can currently do in biological applications. So the tools of biorobotics can really help us challenge our assumptions about how the insect brain might work. So I'm hoping that a research program going forward to explore this is going to be a very intimate fusion of computational neuroscience and experimental neuroscience. I do think there'll be payback, though, for biorobotics and autonomous robotics as it develops into the future. Because we know, and it's talked about a lot, about how the deep learning methods and the machine learning methods are not yielding the outcomes that we might want from autonomous robotics. And Stefan Stelz-Achmacher, who was the CEO of Starsky Robotics, which is a company trying to develop autonomous trucks, and I believe that company kind of failed. And I believe they've been quite honest in saying the reason they failed is because they were leaning on deep learning, machine learning approaches, and the consequence of leaning on those approaches is they realized the limitations of those approaches. Our expectations are up here, our reality asymptote out at here, and we don't seem to be able to break that asymptote with those approaches. And in articles that Stelz-Achmacher has written, he talks about, well, to crack this, we've got to give them true intelligence rather than machine learning intelligence. I think that's wrong. I think to crack this, what you need to do is look at organisms that have achieved functional autonomy and effective autonomy, like the honeybee, and then work out how a honeybee has achieved functional autonomy, and then implement that solution. So let's look at a simple organism with a very small brain that has basically exceeded what your truck can do, work out how it's doing it, and then implement it. So I think that this approach could be very productive for robotics as it goes forward. So to conclude and to come back to the question that posed this, Vincent de Chia was asking, is an insect robotic or is there someone inside? And what I've realized, I think, and what I'm comfortable saying now, I think there is something it's like to be a bee. I think a bee is experiential, but that something is not like me, not by any chance. We've got to become much more sophisticated in the way we think about conscious phenomena. I think it's fair to say a bee has a minimal kind of consciousness. But I think what's really beautiful about this is I think work on organisms like bees is going to help us understand that minimal type of consciousness and going to give us an experimental way, a novel experimental way to tackle the neuroscience of consciousness. And I think that is a wonderful hope for how we could progress neuroscience into the future. And I'll end there. I acknowledge the funding agencies who've contributed to this work, and thank you for your attention.